Hello everyone, welcome back to my channel Microbiology Easy Notes. If you like this channel, please like, subscribe, comment and share. Today's topic, oxidase test. The oxidase test is a biochemical test used in microbiology lab to detect cytochrome C oxidase enzyme in bacteria. The test helps in initial characterization of gram-negative bacteria. Cytochrome C oxidase is usually present in bacterial cell membrane, and this enzyme involved in the electron transport chain, which is a crucial part of cellular aerobic respiration. Cytochrome C oxidase accepts electron from cytochrome C and transfers them to the oxygen. Cytochrome C is a small iron-containing protein. It is present in bacterial periplasm. What happens in electron transport chain? Electrons from NADH and FADH2 pass through complex 1 and 2. Electrons move to complex 3, then transfer to cytochrome C. Cytochrome C carries the electrons to complex 4, which is a cytochrome C oxidase. At complex 4, electrons combine with oxygen and protons to form water, a crucial step in energy production. Oxidase test is first introduced by Gordon and MacLeod in 1928 to distinguish Neisseria gonorrhoeae from Staphylococcus species and Streptococcus species. Later, Kovacs modified the test and used tetramethylparaphenylenediamine dichloride for the identification of the cytochrome oxidase enzyme. This reagent, also known as Kovacs oxidase reagent. Again, Gaby and Hadley modified the test and used para-aminodimethylene oxalate with alpha-naphthalol as a reagent to detect the cytochrome oxidase enzyme in tube culture. How the oxidase test works. A reagent such as tetramethylparaphenylenediamine dihydrochloride is used. This compound acts as an artificial electron donor. If cytochrome C oxidase is present, it will oxidize the reagent. This oxidation causes a color change, typically from colorless to dark purple-blue within 10 to 30 seconds. Reagents required for oxidase test. Oxidase reagent, which is 1% tetramethylparaphenylenediamine dihydrochloride, filter paper or oxidase test strips, sterile loop or applicator stick, bacterial culture not older than 24 hours. Procedure for oxidase test. Moisten a piece of filter paper with a few drops of oxidase reagent using a sterile loop or wooden stick. Pick a small amount of fresh bacterial colony from the agar plate. Smear the bacteria onto the moistened filter paper. Watch for a color change within 10 to 30 seconds. Result interpretation. If after 10 to 30 seconds, dark purple or blue color appears, it means cytochrome C oxidase is present and test is positive. If there is no color change, it means cytochrome C oxidase enzyme is absent and test is negative. And here is some examples of bacteria which gives oxidase positive and negative results. Oxidase positive bacteria are Pseudomonas, Neisseria and Vibrio, and oxidase negative bacteria is E. coli. There are some variation of oxidase tests. In dry filter paper method, a drop of oxidase reagent is added to filter paper. This is a simple, quick screening method. In direct plate method, reagent is applied directly to colonies growing on agar. In swab method, a cotton swab moistened with reagent is rubbed on a colony. This is easy for clinical or field work. Test strip method, reagent is embedded in paper strip that change color when oxidase positive bacteria are applied. In liquid culture method, reagent is added to liquid culture and a color change is observed. This test is useful in specialized labs. Microplate method, high throughput test using microtiter plates and a reagent added to many strains in parallel. This test is useful in research or industrial labs.